Okay, hi everyone. We're going to get started with our 3-minute thesis competition. Welcome, thanks for coming. My name's John McNamara. I'm involved in our conference organizing committee. Thanks for coming and supporting our competitors. This is our first year that we are running our 3-minute thesis competition as part of our Mapping New Knowledges conference. So I'm really happy that you're here. I'll give you a bit of background to our 3-minute thesis competition. This idea was born in Australia a few years ago, and the idea was that graduate students presented their research in 3 minutes. And essentially, the idea was to talk about what you did and how it could change the lives of the people in the audience. So the talks are not particularly academic, they're for a broad audience, and we have exactly 3 minutes to present this work. Today we have 6 competitors who I'll introduce, and they are competing. They're actually competing for a spot in the provincial competition that's going to be held next Thursday at Queen's University. So we have a winner today and a runner-up, and the winner is going to be awarded with $500, which is nice, and they will also be traveling to Queen's next Thursday to represent Brock. We think there are about 30 some odd competitors from the province next week who are competing, and about 17 schools. So it's an exciting opportunity for one of our 6. We have a panel of judges with us today who are going to be judging our 3 minutes. We have Doug Herod from the St. Catharines Standard. We have Kathy Maiteni from Research Communications at Brock, and Jeff Sinibaldi, Manager of Media Relations at Brock as well. Our judging table is up here. They're going to be judging on 3 different criteria. I encourage you to cheer on your fellow mates up here as they present. Each of our presenters, here's our 6. I'll present them individually as they come up. The rules for 3MT are that you have 1 static PowerPoint slide that you can present. So that's what you're going to see today. You're going to see 1 slide for each of our competitors. We have a timer up here. We won't be as hard as they are at the province, but at Queen's next week, 1 second over 3 minutes equals disqualification. All of our competitors are in the latter stage of their thesis research, so most of their data is complete. That's part of the eligibility criteria also, and I encourage you to think about this because we'll have this next year as well. So without further ado, I'm going to hand it over to our first competitor, Abdullah, and we'll switch to his slide.
Thank you all for coming today. But I trust that almost all of us either drove here or took the bus. Either way, you have been burning irreplaceable fossil fuel and emitting carbon dioxide into the atmosphere. Over the past few decades, due to human activity, carbon dioxide levels in our atmosphere have been rising at an alarming rate. This has caused irreparable damage to both aquatic and terrestrial ecosystems. Within the next 50 years, we will have no more coral reefs left on this planet unless serious action is taken. Every second of every day, the sun gives us enough energy to satisfy our needs for the next 8 years. Plants, algae, and cyanobacteria make use of this. They have intricate and complex protein networks that function like nano machines. They take electrons from water, turning it into oxygen, and then shuttle those electrons to eventually result into production of something called ATP. It's fuel for the cell, for a living cell. You cannot run a car with it. Human attempts to replicate this suffer from what I call a forbidden trinity of cost, reliability, and efficiency. Basically, if it is reliable and efficient, it's nowhere near cost effective. If it's cost effective and reliable, it's nowhere near efficient enough. In my research, we have basically taken all the lessons that we have learned from studying natural photosynthesis to come up with our own version of photosynthesis. We use very stable proteins to solve the problem of reliability. They're very reliable because they're stable. We use proteins that are produced in a bacteria called E. coli. This bacteria is called the workhorse of molecular biology because we can make its proteins on an industrial scale. An example is all the insulin that to treat type 1 diabetes today. Our bacteria will take water, turn it into oxygen, and shuttle those electrons over to a protein that takes carbon dioxide, the bad carbon dioxide we emit, and turns it into a fuel that we can put in our cars, formic acid. You always hear about carbon neutrality. Well, this is carbon neutrality at its best. One molecule of carbon dioxide makes one molecule of fuel. That molecule of fuel burns to make one molecule of carbon dioxide, hopefully stopping this devastating trend that is catastrophizing our planet. Well, this is an industrially viable solution. We can actually make this on an industrial scale. It's cost effective. And most importantly, it helps us answer a higher call, a moral obligation to this planet and all the species that we share it with. Thank you very much for your time.